all for coming. I'm just going to do the housekeeping announcements and filling in for Professor Burley, even though I don't have the bow tie. Professor Darren York will introduce our speaker today. But before that, I just wanted to announce that this is the second to last seminar of the series. And the next one is, if you go to the next slide, is by Romy Amaro from UC San Diego. The important thing to note is that this seminar is going to be virtual only. So, and the Zoom link is here, and I'm sure you will receive it via email as well. And this is the last seminar for this semester. So those are the housekeeping announcements. Thank you again for coming. And without further ado, Darren.
Hey everyone, I am absolutely delighted to introduce Fiorella Prada from the Department of Marine and Coastal Sciences. Dr. Prada received her Bachelor of Science in Natural Sciences and Master's in Ecology at the University of Parma in Italy, and earned her PhD in Biodiversity and Evolution at the University of Bologna. Dr. Prada went on to do postdoctoral work at University of Bologna in coral ecology and biology, including studies of temperature, pH, and cumulative human impact effects, and analysis of photosynthetic efficiency. The Department of Marine and Coastal Sciences at Rutgers was able to recruit Dr. Prada in 2021, and since then she is engaged in studies on how biomineralization processes work in corals, foraminifera, and coccolithophores, and might provide new insights into the understanding how the environment has shaped the geological signatures we use to interpret potential trajectories on the planet. So I'm delighted today to introduce her. She'll be giving us a presentation on her work using shallow water hydrothermal vents as natural laboratories to study climate change impacts on corals. Her presentation is titled Natural Acidification Drives Morphological Plasticity of Coral Skeletons. It's a pleasure.
Thank you very much for being here. I'm extremely honored to have had the opportunity to be here today and really share a little bit with you about my research, about my career path that brought me here, and a little bit also about myself. So let's start with yes, this is not like this. Okay, I'm just gonna do it here. So I was born in Costa Rica a while ago. And I started traveling the world with my parents when I was very, very young. So before I was even 3 years old, we moved to Mozambique. And the reason I'm telling you this is because this was a really life-changing experience for me, because it really introduced me to what will become the environment that I will want to investigate and study for the rest of my life. So I will forever be grateful for this experience. Right after this African adventure, we were transferred to New York City, ironically enough, which is pretty funny how I ended up working pretty close by as a professional. And it also was an incredible opportunity to be around people from all over the world. I ended up going to the International School of the United Nations, which had kids from more than 50 nationalities. So it really helped shape also my personality and my social skills. So diversity was really the key word of this experience. And then I moved to Italy, where I really started my academic path. So I started working on the impact of climate change on coral communities. I did my master's thesis and my PhD on the impacts of increasing temperature on population dynamics and growth in Mediterranean corals. I always knew I wanted to combine my passion for the field and for diving with science. And so I was able to do this by working with a research group in Bologna that was doing a lot of field work along the Italian coast. So we were able to sample coral communities along almost 1,000 kilometers of coastline, which pretty much covered a temperature difference of 2 degrees Celsius. So we basically used this latitudinal temperature gradient as a natural laboratory to study the effects of temperature on these communities. And then I entered into the ocean acidification world, using again a natural laboratory to do these studies. And this will be the focus of my talk today. I will talk about this in a few slides. But before I go there, I want to say that it was 3 years ago I had the opportunity to come to Rutgers and work with Paul Falkowski at the Department of Marine and Coastal Sciences on biomineralization. So while I had been doing coral ecology work for 10 years, I then moved into more molecular work on biomineralization and on the mechanisms behind this process. And this was extremely exciting for me because it really allowed me to try and understand some of the mechanisms behind the observations that I could see in the field. And so eventually this postdoc became a position, an assistant professor position a year ago. So marine calcifiers have really been always the focus of my work. These organisms are crucial organisms in the ocean. They build calcium carbonate structures from calcium carbonate and or bicarbonate ions and form shells or skeletons. They are fundamental players in shaping our planet by providing crucial ecosystem and biogeochemical functions. So for instance, some of these organisms are called ecosystem engineers, and reef building corals are a perfect example of this because they build these fascinating structures that you see here also in the background, which form the habitat for hundreds of thousands of species that depend on these ecosystems for a whole range of parameters or features, such as nursery, protection, food. In fact, these ecosystems are a source of food, not just for the direct inhabitants of these ecosystems, but also for those millions, and literally there's 500 to a million people that live along coastal areas that depend almost exclusively on these ecosystems for food. But marine calcifiers are also crucial for global elemental cycling, as they regulate the carbon cycle, the calcium, the nitrogen cycle, for example, through processes like carbon sequestration, nutrient cycling, and ocean alkalinity buffering. So ultimately, calcifiers, through their activities, support marine biodiversity. They sustain food webs and contribute to the long-term carbon storage, making them essential for ecosystem stability and climate regulation. So let's look at a few examples of these calcifiers, and specifically, I will talk to you about the three main groups of calcifiers that contribute significantly to the global carbon budget. So we have stony corals that we already mentioned, which are also called hard or scleractinian corals. We have single-celled algae called coccolithophores that build these fascinating structures. They're called liths. These are basically calcium carbonate plates that have very articulate morphologies and form these so-called cocospheres and are a very important part of the phytoplankton community. And then we have foraminifera, which are single-celled protists. These also build calcium carbonate shells, and both foraminifera and coccolithophores are very important for in geochemical studies, because by investigating the isotopic signatures of their shells in the fossil record, we can trace back and understand and study what the conditions of the ocean was millions and millions of years ago. So the contribution of these three groups, only these three groups of calcifiers to the global carbon budget has been estimated to be around 3.5 gigatons of calcium carbonate per year, which corresponds roughly to 600 Giza pyramids. So through the formation, sinking, and dissolution of this incredible amount of calcium carbonate, these organisms play a crucial role in regulating marine carbon cycling and also ocean atmosphere CO2 exchange, which means that they ultimately really influence our climate. So, well, on one hand, we know that they influence our climate. We also know that they're very, very sensitive even to the slightest changes in environmental parameters most of the time, as we are seeing, for example, for corals globally. We also know that these organisms have been around for millions of years, though. So it's how do we solve this apparent paradox? How is it possible that these crucial organisms that are so sensitive to environmental change manage to survive millions of years of global climate change? So this is really where my research question stems from. So I am interested in studying how marine calcifiers are affected by the changing environment, and specifically corals, and how the environment in turn affects them. Whether the conditions are healthy or also under unhealthy conditions. And so why corals though? Well, there are a million reasons to investigate these fascinating creatures. First of all, they have an immense ecological value, as I already mentioned. They are responsible for building these magnificent structures, not just in the surface. In fact, all of us know and appreciate the beauty of tropical coral reefs, but as we will see in a few slides, these organisms can build these fascinating ecosystems also in other environments, such as the deep sea. So they can build literally forests of calcium carbonate at 3,000 meters depth in complete darkness and at temperatures that are super low, around 3 to 4 degrees Celsius. We still don't fully understand how this works, of course, but this is the beauty also of working with corals. There's still a lot of unknowns. In any case, regardless of where they build these environments, these organisms provide a lot of ecosystem services, as we said. So in terms of when we talk about tropical coral reefs, of course, they sustain entire fisheries. And they not only do they sustain the economy of these areas, but they also protect them from bioerosion and from storms. So they're really important. Corals, however, are fascinating creatures also because of their amazing ability to overcome dramatic environmental change, as witnessed by their incredible history of survival and evolution throughout hundreds of millions of years of global climate change, as you can see here in this slide. So here on the top panel, you can see the global ocean surface pH in pink. Please note that the axis has been inverted so that you have the increasing degree of stressor growing from the bottom to the top of the plot. And you have sea surface temperature in light blue. On the bottom, you have the distribution of corals, of rebuilding corals in the past 250 million years. The reason why it's plotted against 250 million years is because this is 250 million years ago is when modern reef building corals evolved, at least to the best of our knowledge, even though there is evidence from molecular clock analysis that corals might have evolved all the way back to 540 million years ago, so a long time ago. So what I would like you to take home from this slide is that over at least, even just looking at the past 250 million years ago, these organisms managed to survive at least 3 major mass extinction events, which were driven by dramatic volcanic activity, in some cases also by abrupt phenomena such as asteroid impacts, and also sudden phenomena like the Paleocene-Eocene thermal maximum 56 million years ago, which according to pretty recent literature seems to have occurred within only a few hundred years and led to increasing global surface temperatures and oceanic temperatures and increasing ocean acidification in a very similar mechanism to climate change today. So of course, it is very hard to compare current rates to past rates because we can't really disentangle the magnitude, yes, but the rate at which these phenomena occurred in the past is hard to really determine with certainty. But the truth is that they survived. They're still here today. And so they have the ability to adapt and to respond to really, really dramatic changes in the environment. So what is their key? What is their weapon? And will they be able to use this also in the future in what has been, unfortunately, increasingly referred to as the sixth mass extinction event? So one of their secret weapons, at least for the shallow water coral species, is their symbiosis with dinoflagellates. Single-celled algae that can transform light energy into chemical energy in the form of carbohydrates, lipids, and polysaccharide, and amino acids that are transferred from the alga to the host in exchange for shelter and for inorganic compounds, which are basically the metabolites of the coral that are recycled by the alga. This is a very important mutualistic symbiosis and it really witnesses the ability of these organisms to make the best out of the resources they have, because these symbiosis are the key to their success in tropical environments generally. This is really where we have its maximum expression. And tropical environments, by definition, are extremely low in nutrients. They're highly oligotrophic, which means that there's very little food. And so what there is a lot of, though, is solar irradiance. And so this symbiosis allows them to fix carbon and get nutrition from the alga in an environment where they would have a very hard time finding food. And so this is really proof of their plasticity and of their ability to try to adapt to a possibly difficult environment for several different reasons, including, for example, in the deep sea. So we think that a similar mechanism might be happening also in the deep dark sea, where there's very little food. And in that case, probably the symbiosis is with some kind of bacteria, for example, chemotrophic bacteria, for example. So just to give you a very quick idea of the diversity and the diversity of coral communities that we have. Corals, and this is a big mistake that is written everywhere. Corals are not just tropical, and they're not just colonial, and they're not just symbiotic. So these are the corals that you see in this slide that we generally see in all the movies, in all the pictures in tropical environments. So this is what we think is out there, and that's it, but that's not true. We have various morphologies. We have these massive morphologies that look like cushions. We have these branching morphologies here on the right. So species with different kinds of morphologies, but the same species can also show an incredible variety of morphology. So this is a typical Mediterranean coral. Here on the left, you can see these massive colonies, which are huge, literally huge, and they form these carpets, beautiful areas in the rocky substrate of Mediterranean waters. And so on the surface, they form these plateaus, basically, of very thick colonies. And the same species, if you dive down to 20, 25 meters, looks completely different. So it builds these sort of branching morphologies. And the reason is because on the surface, we have a lot of hydrodynamism, a lot of water energy. And so they need to be stronger and thicker. On the bottom, you don't need that. And so they transform completely their morphology. We also have solitary species. So well, in this case, these colonies are made up of sometimes hundreds of thousands of polyps, which are the single animals that are genetically identical to each other. So all of these are from a pioneering polyp that buds and creates the colony. There are also species that are single polyps and are very widely distributed in temperate areas, for example, and in the deep sea also. And then we have two different kinds of energy intake strategies. So we have the non-zooxanthellate corals, which are purely heterotrophic, so they feed exclusively by capturing food from the water column. and particles and organic matter in general. And then we have the zooxanthid corals. Zooxanthid because these dinoflagellates are called zooxanthellae, so this is where they take their name from. And so they depend in many cases almost exclusively on the symbiosis. In fact, 95% of the carbon fixed by the alga is transferred to the coral. So all of these flavors of corals we have basically form many different kinds of coral reef ecosystems in a wide array of environments. So tropical environments like you can see here, temperate environments like you see here. This is a picture taken in Croatia. So this is Cladocora cespitosa. This is the only reef building coral found in the Mediterranean Sea. And it literally covers entire patches of seabed sometimes. And even though, of course, it's less it's less colorful and not as mind-blowing as you can see in tropical ecosystems, but and it's monospecific to some degree. Still, this species is able to create these habitats that sustain many different species. So the mechanism is always the same. And the same thing also happens in the deep sea. So this one is the main deep sea coral reef builder called Lophilia pertusa or Desmophyllum pertusum. It recently changed its name. And as you can see, it can literally form forests of these branches that cover different like kilometers sometimes of seabed, and obviously creating a lot of habitat for many species again. And what's fascinating about this coral in particular is that despite growing up to 2,000 or to 3,000 meters, it can still build really thick and really strong skeletons. So they're super dense, super hard to break. And we don't really understand how, like where they really get their energy, because almost all of the organic matter that falls from the surface to the seabed gets consumed in the first 1,000 meters. Only less than 10% is able to go beyond 1,000 meters depth. So the amount of food that they get from the surface is very limited. So we still need to understand how they survive, but they do, and they survive. They thrive really well. So just to give you a very brief explanation of what a coral is, so it's easier to understand what we're going to see later. So here on the left, you can see a typical colonial zooxanthid tropical coral, Stylophora pistillata. It's basically the lab rat for coral reef, for coral ecologists and biologists. So if we zoom in into one of these branches, we can really see that it's made of hundreds of thousands of tiny polyps, as I was always showing earlier. And these single polyps are connected to each other by a very thin layer. So the way the coral skeleton is made is you have literally the structure that you see here. This is the skeleton, just covered by a thin layer of tissue. And the skeleton, it's not smooth. It actually has these cup-like structures that I tried to draw here in this picture up here. So these cup-like structures basically sustain the single polyps, sort of like a beaker or a glass would hold a glove if you placed it on top of it, with the fingers being basically the tentacles. And so if you look at a cross section of the coral tissue, you can see that it's actually, there's several different layers that separate the external seawater environment from the skeleton here on the bottom. And which means that the skeleton is not in direct contact with what is happening outside. Moreover, these tissue layers, basically the last tissue layer that we have before the skeleton, it's a very specialized tissue. It's made up of cells called calicoblastic cells that are responsible for the precipitation of the calcium carbonate skeleton. And they are separated from the skeleton by a very thin fluid. It's only a few nanometers, so a couple of microns wide, which is called the calcifying fluid, which is really where the calcification reactions take place. So everything that is responsible for producing the skeleton happens down here, which is pretty, I mean, pretty separated in a way, not completely, but to a good degree from the external seawater environment. So let's just take a really quick look at this fluid. So the composition of this fluid is both passively and actively controlled. So passively controlled by paracellular pathways and actively through transcellular pathways. We also know that its composition and pH, for example, can be very, very different from that of the external seawater environment. In fact, when outside, you can have a pH of 8.1 or even below that, 7.8, for example. In the fluid, you can have really high pHs of 8.7, 8.9, which is a huge difference, which means that the organism really exerts a very strong biological control over this so-called privileged space. And this is accomplished through the activity of several proteins. So we have, for example, calcium ATPase pumps that pump calcium into this fluid, and in exchange for protons that are expelled outside of this fluid. And this allows the maintenance of high pH in here. But we also have the activity of other proteins which are part of the so-called organic matrix that is produced and secreted by these calicoblastic cells into the fluid. So this organic matrix, it's made up of lipids, proteins, and polysaccharides. And they sort of form a template that induces the precipitation of the calcium carbonate. So when this organic matrix is secreted, it creates this template that is basically incorporated in the growing skeleton layer by layer. So corals literally grow one layer after the other, and you can literally see this also when you take CT scans of the skeletons, for example. And so if we want to study what is happening, so the bioionization process, for example, and this is exactly what we did when we try to sequence these proteins, is extract this organic matrix and then look at the composition and function of the proteins inside. And this allows us to sort of get time-lapse images, let's say, of the bioionization process and try to understand how it works. So I'm telling you all of this because the take-home message here is that the bioionization process is very complex and it's strictly controlled by the organism, which means that when we study processes like ocean acidification, for example, that involves the absorption of CO2 into the ocean, which leads to increased protons and so decreased pH. But these protons also end up combining with carbonate ions that are available in the seawater, forming bicarbonate ions. So since carbonate ions are the main building block used by these organisms to build their structures, this means that acidification has a dual negative effect. So on one hand, it can lead to the dissolution of structures that are already formed because the pH can go very, very low in some cases. But it also makes it harder for organisms to build new structures because it reduces the availability of carbonate ions because they combine with protons that these organisms use as building blocks. Of course, it's much more complex than this, but this is pretty much what the summary of what has been going on and what is discussed constantly in several scientific forums and literature. So all of this, however, doesn't necessarily translate into negative effects in biomineralization because of the strict control that is exerted by the organism. And this is what explains the wide array of responses we see in many studies, in which some cases organisms start biomineralizing even more when you have higher acidity. Sometimes they don't respond at all. They don't change their bioionization. So there's still much to learn. And we certainly need to start from learning the mechanisms of bioionization, which we still have very little knowledge of in many cases. So how do we study this? How do we study the impact of acidification? One way to do it, and this is most of the studies do it this way, is to use controlled environments, so aquarium systems, for example. This is a very fancy facility that was built in Eilat at the Inter-University Institute for Marine Sciences, where they built these sets of aquaria that are controlled automatically by this robot here in the middle. In the back, you can see the Red Sea, so it's beautiful. And the water, of course, is provided directly from the Red Sea. So you can literally control many different parameters and see how a single parameter or two parameters in combination affect several features in various organisms, not necessarily corals. But this doesn't really mimic what is happening out in the environment. There are a lot of things that you cannot mimic, like currents, nutrients, seasonality, for example. And so one way to do it with, of course, there are a series of caveats because you cannot control everything outside in the environment, but that's also the positive side of it, is to use natural laboratories to do this. And CO2 vents, which are basically, they are hydrothermal emissions coming from the seabed. They're generally of volcanic origin. And what happens is that some of these CO2 vents are shallow enough that we can study them. We can dive there and literally study them. And they also need to be at ambient temperature because the problem with many of these hydrothermal emissions is that they're extremely hot. But some of these actually manage to get out of the seabed basically after being cooled down through the crevices of the crust. And so when this happens, these emissions come out at the same temperature that the external seawater is. So this is a very important feature. And then also they have to lack toxic compounds. So I was lucky enough to work at one of these sites. It's located here in the Mediterranean Sea, specifically here. This is a volcanic arc of islands. It's called Aeolian Archipelago. And one of these islands here, I mean, there are several vents throughout all this area, but one of these vents was the one that we worked on. And the reason is because this is a this is a crater, an underwater crater at more or less 10 to 12 meters depth. So I'm sorry, I can never do the conversion in feet, but it's pretty shallow. So it's relatively easy to dive there. As you can see, there's a very important column of bubbles of almost pure CO2 that comes out from the seabed. When you dive on top of it, you can literally hear it, feel it stinging on your skin. And so these bubbles acidify the surrounding environment and create very low pH conditions close to the crater, which are then gradually restored as you dive outside of the crater along this transect. So we conducted several expeditions in this place to really characterize the environment and make sure that this pH gradient, natural pH gradient, was maintained for a relatively good, I mean, at least for an entire year. So we saw that even seasonal changes were not different and did not affect the pH conditions of this gradient. And so we were able to study calcifying organisms, and I will talk about corals, of course, that naturally grow along this gradient. And so for generations, they were exposed to these pH conditions, different pH conditions along this gradient. And these pH conditions basically match different IPCC, Intergovernmental Panel of Climate Change scenarios for pH for the end of the century. So in our control site, which is the furthest one, the last one we saw in the video, we have a normal pH condition. So this was our control. And as we move along the pH gradient, we have values that match an intermediate scenario for the end of the century. And we also have in our third site a pH that matches the business as usual scenario, which is the scenario by which predicted if we continue emitting CO2 at a current rate. And we also have a very extreme pH condition, which is the one predicted for year 2300. So we have the whole spectrum of conditions and to see how they affect these coral communities. I will show you a few more videos of some of the activities we conducted every time we dove there. So here I was collecting pH, temperature, and salinity data right on the crater. So when I was sitting on top of this crater for several minutes, uh, it's really acidic. It really stings on your skin, but it's a very incredible place. It's like being on another planet. But of course, in here in the center of the crater, there's nothing. We decided to characterize it just to have like a complete picture. But the first site that we actually worked on was at the rim of the crater, where we don't have such low pHs. Here we have a pH of 6.4, which is way too acidic. So in addition to the environment, we also took many different physiological measurements. So this is an underwater fluorometer that basically measures the photosynthetic efficiency of the symbiotic alga inside the coral. So by measuring how well the photosynthesis is working in the alga, we can determine the health of the coral. So if the alga is happy, the coral is generally happy. And so this is a non-destructive technique that is really quick and can really give you a general idea, at least, on the physiological condition of the corals along the gradient. And then we also couple those physiological measurements to growth measurements with this very, very technological plastic caliper, as you can see, which we use to get biometric measurements and then get to the growth, which I will show you in a second. So all of these studies basically were conducted. I will show you the next slide. The next slide basically summarizes 10 years of work, which is pretty crazy to be able to show in one single slide. So the idea was to basically investigate the acclimatization, so the ability of this coral to respond or not to respond, or to be negatively affected by these persistent acidified conditions. So we conducted this multi-scale analysis of these populations by investigating from the population scale all the way to the macro, micro, and nano scales. So we did go all the way down to investigating the microbiome composition and the reproduction, the physiology, the physiology, and also all those microstructural features of the skeleton, which is really what I would like to focus my attention on today. So today I will tell you everything that has to do with how the skeletal phenotype responded or did not respond to changing pH conditions. So in the next few slides, I will show you a few results. They're all formatted in the same way. So all the plots will show our control side on the right in blue, and our intermediate side in the middle, and the lowest pH side in red on the left. So as you can see, there are only 3 out of 4 sites here because ultimately this coral species is able to withstand only pH values up to 7.7. So our lowest pH, 7.4, which was the one predicted for year 2300, is not compatible with this coral species. So this was already a pretty clear result. And then, of course, just by diving there, you could see that the population would decrease dramatically. And so we measured this, of course, through photoquadrats and assessed that the population density actually decreased. And to try and understand why it decreased, we investigated coral growth. So coral growth is defined by these 3 parameters, which are net calcification rate, which is the amount of calcium carbonate produced per milliliter square per year, which is the result of linear extension rate, which is how much it grows in length in a year, where the length in this case is considered the maximum axis of the oral pole of the coral. So this is a solitary species. And so the oral pole is basically taking the coral, looking at it from the top and taking the maximum diameter. And then we also measured the skeletal bulk density with another non-destructive technique. So we basically measured these 2 parameters independently to get to the net calcification rate. So linear extension rate was measured by computerized tomography. So we took CT scans of these skeletons. We knew already that they have a seasonal growth, so they always deposit a high density band in the winter, which is when they eat more. and a low density band in the summer. So by counting the number of bands, we can determine the age of the specimen. And by dividing the length of the specimen by its age, we could get to the linear extension rate. So that was a pretty straightforward way to get to this parameter. The skeletal density was obtained by buoyant weight. So again, not a fancy technique, but very effective. So the buoyant weight technique is based on Archimedes' principle, which states that an object immersed in a fluid will displace a volume of fluid that is equivalent to the volume of the submerged object. So based on this very simple the method, basically, we were able to get to these three important skeletal parameters, which are the density of the skeleton and its porosity. In fact, the coral skeleton isn't just a cube of calcium carbonate. It's very articulate and full of pores and cavities and canals that can affect its structural integrity. So and then we also were able to get to the microdensity, which is the density of the material. So 98% of the skeleton, 97, is made up of calcium carbonate, but the remaining part is organics. So the organic matrix I was telling you about is incorporated in the coral skeleton and can affect its mechanical properties. And especially the corals can control the amount of these organics that they produce. And it can tell us really a lot on how they respond to change, to environmental change. So what we saw was that with decreasing pH, there was a significant decrease in the net calcification rate. And this was the result of a decline in the bulk density, in the density of the skeleton, while the linear extension rate was constant. This was a pretty important result because the main parameter that usually coral biologists use to assess coral health is coral growth. And by coral growth, they mean linear extension rate. And so if we had stopped at this parameter, it didn't change. So we would have mistakenly thought that these corals were doing just fine, because the fact that this is constant means that the corals were actually pretty much the same size along the pH gradient. What we wouldn't have known is that something is going on the inside that is really affecting their net calcification rate, which is what really matters. So declines in bulk density are pretty ubiquitous with decreasing pH, also in many other species. So here we pulled together the data from 4 different tropical coral species that were sampled in Papua New Guinea, which is another very, very important place where there are CO2 vents and it's very, very studied. And so we see the same decreasing trend, and the same trend is observed even under control conditions. So this coral species Stylophora that we saw earlier was investigated for a whole year under decreasing pH, and again showed the same decreasing trend. So all of this obviously affects the mechanical properties of the skeleton, which we measured by nanoindentation. And so this is a very used technique in material sciences, and it allowed us to obtain the stiffness, which is the resistance to reversible elastic deformation, and also the hardness, which is a measure of the tendency to break, the tendency of a material to break. So we already knew that stiffness is actually often related to macroscale features. So if bulk density changes, stiffness generally changes. And if porosity changes, stiffness changes. And this is exactly what we saw. So increasing porosity obviously decreased the stiffness. So the skeletons become obviously more fragile, and this was quantified like this. The hardness did not change, and the hardness is generally related to parameters that are more nano to microscale. And so this means that the structural features of the skeletons were pretty much the same. So it seems like the building blocks did not really change. The mineral composition was the same. The amount of organic material was surprisingly the same also. So the organic matrix produced was not different. So we still don't know what's going on. So why does this density decrease? So the next thing to do was to try and investigate the pH at the site of calcification. And we did this by using boron isotopes. So there are 2 dominant species of dissolved boron in the ocean. Boron 10 and boric acid, sorry, which is shown here in blue in this plot, and borate ion in red, and their concentrations are pH dependent. So there are two isotopes of boron, boron 10 and boron 11. By convention, stable isotope ratios are reported as delta values, where which is a means basically of comparing the ratio of the heavier isotope to the ratio of the lighter isotope to that of a standard. And since these changes, these differences are so small, we multiply everything by 1,000, which means the delta values are always in per mil. So we also know that there is a 27 per mil difference in the isotopic difference between these two boron species. And we also know that borate ion is a species that is primarily incorporated in biominerals. So by measuring the isotopic composition of borate, we can calculate the pH at which that biomineral formed by simply looking at this plot. So in collaboration with colleagues from the Geomar Institute in Kiel, Germany, we used laser ablation to take these measurements. And this is what we obtained. So the calcifying fluid pH, shown here on the bottom, was not different. It did not change regardless of external seawater pH, which means that the delta pH, so the difference between the calcifying fluid and the external seawater pH, dramatically changed, of course. And so just to wrap things up, long-term acidified conditions seem to affect these skeletons at the macro scale in terms of density and net calcification rate, but at the nanoscale, we were not able to observe any differences, any specific signals that that could explain this dramatic change in bulk density and calcification. So the question remains open: what is driving these changes? So to try and answer this question, we need to get into the mechanisms of biomineralization again. So biomineralization is, as I said, a biologically controlled process in which the organic matrix, so these lipids, proteins, and polysaccharides, are secreted by these calicoblastic cells shown here in pink that create a template that induces the nucleation of the mineral phase. So several studies, however, have shown that upon secretion of this organic matrix, we actually have the deposition of an intermediate amorphous calcium carbonate phase, which is shown here as these sphere-like structures, which are only barely 100 nanometers wide, which subsequently aggregate and serve as building blocks for constructing the aragonite crystal structure. So basically, this sequence of events translates into a so-called two-step model of coral calcification that I will briefly guide you through. So if we take our colony and we remove all the tissue from the surface, we are left behind with these cup-like structures, which are called corallites. And if we zoom in into one of these corallites, we can see that there are these intersecting structures that you see here, which are called septa. Again, if we zoom in into one of these septa, we can see that they are not actually smooth on the surface. They have these sort of bumpy structures. And if we zoom in even more on the inside, we can see that these structures have a sort of layered morphology on the inside. So this morphology is formed by vertical upward growth of so-called rapid accretion deposits, shown here in light blue, and lateral, which are very, very rich in the organics, inorganic matrix, and lateral growth of aragonite fibers, also called thickening deposits. So we have this mechanism: rapid vertical upward growth and lateral growth. This is the dynamics of the calcification process, which we understand pretty well so far. What we don't really understand well yet is the organics. So what is the composition and the function of this organic material? So there has been a lot of work in the past few years. A lot of proteomic studies have been able to identify many of these proteins. some of which are calcium binding proteins, adhesion proteins, several enzymes. And there's a group of proteins that are acidic, there are acid-rich proteins, which are pretty ubiquitous along across marine calcifiers. That have a pretty significant role in the bionization process because, so they're highly acidic, as you know very well, because they are rich in glutamic and aspartic acid. These two amino acids basically form negatively charged sites along the protein sequence, which strongly bind cations such as calcium. And moreover, these proteins, some of these proteins at least, have been expressed in the lab. So we've been able to express them and to induce them to precipitate calcium carbonate in a test tube, even at pH 7.6. So very, very low, even lower than the lowest pH site we just saw just now. So this really points to the direction that biomineralization is not necessarily strictly related to changing aragonite saturation conditions on the outside. It's more complicated than that. And so this is why it's important to really tackle and understand how this organics works and drives the process. Also, all these proteins, they're not just a toolbox. They're not just a group of proteins working independently. They're actually a factory of proteins working together in interactomes. They form networks of proteins that drive the bionization process. This was shown, and thanks to actually the work of Paul Falkowski and his team, thanks to the amazing facilities here at Rutgers, the mass spec facility that allowed us to determine this network of proteins. And so is all of this only, I mean, obviously all of this is not just a coral thing. There must be some kind of shared pathway across marine calcifiers. And so the idea is really to, the idea of one of the projects I'm working on is to try and understand these mechanisms across these three main groups of calcifiers that we talked about today. So to start off, we have managed to extract and purify proteins from coral skeletons again, this time on the deep sea coral skeletons. We did this in foraminifera and coccolithophores. And we actually just managed to publish the data from the proteomics work on the foraminifera, which has shown that it was an extensive proteomic analysis that showed significant insights into the biochemical complexity of this process in foraminifera. I'm not going to go into the details of this. It would be a completely different presentation, but it really does highlight how proteomics can help us sort of see what these time lapses are of bioionization and try to fill some of these gaps. And so my final thing is, again, open question still on how the mechanical features of calcifiers are guided. So, and how does this organic matrix really what is the role of the organic matrix in determining these mechanical features? So what I really want to do in the next few years is try to investigate coral skeletons or other calcifiers under different conditions, investigate their mechanical features, and use this tool, proteomics, to really understand what the composition is of these proteins and what their function is, and if there are any correlations between the protein compositions and functions with the mechanical features that we see in these skeletons, and try to understand what drives porosity, for example, in coral skeletons. We have no idea. And this has, I guess, several applications also in many other disciplines, such as biomaterials and things like this. So all of this was made possible thanks to the collaboration with amazing people, which I chose not to list the names of everybody, but just show the institutions that are distributed pretty much all over the place. And so I like to show this slide, especially to students usually, because it really highlights how much our work is interdisciplinary and international. So it's super important to collaborate with people with different expertise and also to share our research with a crowd of people like you today, for me, that work on widely different topics, but that can have insights that I would never have because we just come from different backgrounds. So interdisciplinarity, I think, is really the key to try and answer some of these outstanding questions. So thank you very much for inviting me here today. It was really an honor for me to be here. Thank you.
Couple questions?
Thank you. That was really amazing. So about, I totally agree with the last topic, that you mentioned about the importance and the relevance of this work to many fields. Could you say a little bit more about the biomaterials aspects?
Yes.
And what kind of, enabling technology this could be or insights this could provide? Because I'm very interested in that. So thank you.
So yes, so coral skeletons have been used and are studied also in biomedicine, for example, as bone graft, for example, for implants and stuff like that. But something fascinating that I found out a while ago was that it can also be used for brain injuries. So apparently, if you use coral skeletal material and implant it into the brain, I guess the porosity of the skeleton, and I don't know if it has anything to do with the organics in the skeleton for some reason, but it favors the growth, the established, I mean the growth of neurons around the substrate, and it also reduces significantly the rejection by the organism because it sort of like recognizes it as a biomaterial, and so it just makes biocompatibility easier for some reason. So I don't know. There's a lot to understand there. It would be very interesting to see if it's just a matter of the structure of the material, so the porosity, or maybe even the organics that somehow favors, I don't know why, chemically or biologically, the attachment of the neurons to the skeleton, for example. So this is one aspect. Yeah.
In the back, and then I'll get to you.
Thank you very much.
Thank you.
Is there a equilibrium process in the growth of the structure? So you showed the finger-like growths. Is that an equilibrium process that can grow and then under certain right conditions, equilibrium might favor dissolution and then regrowth? And sort of a corollary question, especially if that's not an equilibrium process and if the coral just forms and it stays there, is a lot of that carbonate uptake then driven by sort of a Le Chatelier effect on the carbonate equilibrium, such that all the carbonate just keeps getting drained because these proteins promote calcification.
Yeah.
And maybe are they just driving the equilibrium such that even under low pH conditions, you just keep generating more carbonate from the equilibrium because it's all getting sucked into the precipitate.
Yeah, so this is something that is actually observed in foraminifera. So you do have the precipitation process there is really favored by the CO2 gradient that is formed from outside to inside. So these proton pumps basically generate a gradient of CO2 that is able to penetrate through the cell membranes and go into the calcifying fluid. So once it gets there, you have proteins such as carbonic anhydrases that catalyze the reverse conversion of CO2 to bicarbonate, and then you have the formation from that bicarbonate. You have the pumps that pump protons out of the fluid, and therefore favor the formation from that bicarbonate of carbonate ion, and therefore calcification. So yes, there is for sure a strong control on sort of maintaining that pH buffering activity in the fluid, which is very energy intensive, by the way. So it's really what what late research has shown is that it's not so much the fact that there is reduced availability of these carbonate ions in the seawater that is the problem. It's the fact that when there are a lot of protons in the seawater, it becomes increasingly more difficult for the organism to expel those protons from the fluid and favor an alkaline environment that favors the precipitation process. And so this is one thing. The other thing in terms of dissolution, so dissolution is really a problem only when the coral skeleton is exposed to the external seawater environment. So when the coral is doing okay and it's completely covered in tissue, as we saw in the first few slides, the tissue really protects the calcifying fluid from what is going on in the seawater. So if the fluid and the skeleton, so it doesn't allow a direct contact between that low pH water and the skeleton itself. So it has been shown actually that one response of corals to acidification is to thicken their tissue. So they tend to literally make it thicker and so more, they tend to try and isolate that space more from the external seawater environment to avoid this effect. Dissolution, though, is an effect that we see in bare skeletons. So it is still a problem because as corals grow and eventually leave dead skeleton behind, because that's how a coral reef grows. You have the dead old skeleton in the bottom and you can have continuous growth on the surface. So that skeleton does get exposed eventually to acidic seawaters and can really, which can and really like start dissolving and affecting the structural integrity of the reef, which ultimately could break down anyway. So there's a lot of counterbalance things there. And also, obviously, acidification is not happening alone. You have increasing temperatures. Increasing temperatures favor the tissue apoptosis. So loss of tissue, which therefore leaves bare skeleton exposed to acidic waters. And so that's where you have the negative and synergistic effect of the two phenomena, which is more important.
Maybe we'd have time for one more question.
Very quick question. Maybe this is difficult to study, but have people looked to see how acidification at the surface layers is affecting the deep water non-photosynthetic corals?
Mm-hmm. Yeah, so it is a big topic, especially because deep waters, of course, deep waters are already almost undersaturated, which means that if we have global acidification, those are going to be the first areas in our oceans to go below saturation. Despite that, the fact that deep sea corals have adapted to living under almost undersaturated conditions for generations, for millions of years, literally, has allowed them somehow to find a way to like counterbalance the effect of these almost undersaturated waters, because we don't understand still why, but we know for certain that the calcifying fluid of deep sea corals is much, much higher in pH compared to shallow water corals, for example. So if shallow water corals have 8.4, deep sea corals can have 9.5 pH in the fluid. And so this is pretty fascinating considering that it's hard. I mean, it's very energy intensive to maintain such a high pH. And so this means that they're already consuming a lot of energy to maintain that pH high. But on the other hand, it has allowed them to already sort of find a way to counteract the effects of acidification. So they tend to be pretty resistant. They tend to sometimes to not show any negative response, while the shallow water corals seem to be less resilient to this, for sure. So one possibility is that deep sea corals could be a refuge environment in the future, because it's more isolated also in terms of temperatures, for example. And so maybe only those deep communities will manage to survive in the future. That's one hypothesis that has been put out there.
So maybe we'll take the rest of the questions from students for a nice lunch upstairs. But before going, we'd like to present our tokens of our appreciation for you coming and giving us a talk. So we have a couple of things. We have a report for you to read That's
A
the
rep-
boring part.
Have
Now
homework?
Now, yeah,
Okay.
exactly. The quiz next seminar that you give. We also have a 2025 calendar
Oh,
to
bones, I
give
like
you,
this.
and most importantly,
Thank you. Structural biology, nutrition.
we have, I'll let you unwrap it.
Yes.
So we have
Oh,
5.
oh, this would have been useful this morning.
Yes.
Nice. Thank you. This is.
So you can
wonderful.
use
Thank you so much.
that for
I
next
love it.
time, and it should have a good lifetime. It's a nice
It's
thermal cup,
It's
so.
wonderful. Thank you.
Anyway, let's give a warm round of applause and
Thank you.
thanks for a wonderful talk. And thank you everyone for coming.